I want to reflect on where we are at the moment, and to start at the beginning. So 50 years ago, monoclonal antibodies were invented by Milstein and Kohler, who won the Nobel Prize for it. And you can see here the tremendous growth of monoclonal antibodies therapeutically since then over that time. You can see bispecific antibodies at the bottom right-hand in the corner. What aren't included here are trispecific and tetraspecific antibodies, which are also in clinical development at the moment. So if you just imagine for a moment, you take bevacizumab, pembrolizumab, trastuzumab deruxtecan, and sacituzumab govitecan come together and have a baby. That's the kind of level of targeting that you're dealing with these drugs. These drugs are dominating our clinical landscape at the moment. These are the 15 trials selected by one opinion in ASCO. Over half of the trials presented at ASCO here as among the top 15 trials have monoclonal antibodies at their heart. And on the right-hand side, you can see the presentations of new antibody-drug conjugates at ASCO. These drugs have never been presented at ASCO before. At the present time, 160 antibody-drug conjugates are under development in lung cancer alone. But these developments need to be tempered with reality. So this lady had a history of early stage breast cancer and received a chemotherapy and immunotherapy on this keynote regimen that you see here. So you can see that she got chemotherapy and pembrolizumab therapy for 6 months, and then she had surgery. And then the pembrolizumab was continued after the surgery, regardless of the results of the surgery. So she presented to the clinic and had noticed the development of craters involving her arm on the side of her breast cancer surgery. And then these craters extended to her other arm, and then they extended to her legs and to her buttocks. And so you can see here the timeline of symptomatology developing here. So skin biopsy showed necrosis and confirming a clinical diagnosis of lipodystrophy. So fat destruction and immune destruction of her fat cells. She was put on steroids as a measure, and this position is stabilized and gradually slowly improved, but the recovery is not guaranteed. And the case really brings to mind the human cost of non-selected, non-personalized therapy in this case. So I guess the question is why the treatment wasn't adjusted after the result of surgery. She had a complete response to treatment, so really why was the treatment continued beyond that? So this is a look at the FDA randomized trials over a 20-year period, and you can see the tendency in these trials has been to escalate, to increase with each one. And you can see in blue at the top, there are two outlier trials, de-escalation trials that went to the FDA for approval. One of them was the BOLT trial in patients with basal cell cancer, which is therapeutically quite rare because most of them are dealt with by surgery. And the other was in prostate cancer, a treatment where a dose of a standard drug, carbazitaxel, was de-escalated. But overall, the majority of trials are in the escalation mode. And part of that is that companies are risk averse. If you're doing a trial and the trial fails, the drug fails, it costs the company can cost up to $50 billion. And the company has a duty of care to shareholders. This has been discussed in both the first session and earlier on by Benjamin also. The complementary roles of academic and industry trials. So as Jerry Hanna outlined in the first slide that Jerushka showed, academic trials address unmet need and optimize what is the approved drug in industry trials. Many of the studies presented by Benjamin were academic investigator-initiated trials. But if the drug is approved, why do we need to change the approval? Surely our agencies have established that this is the correct dose and we move on. Well, the first thing is the trial population may not be representative. You can have age, comorbidities. There may be emerging evidence, new biomarkers that might modify the treatment that's given. And also there's affordability. On the right-hand side, you can see that the implications of de-escalating would be less side effects for patients, as Benjamin has showed, reduced financial toxicity, as he's very clearly showed, reduced time toxicity, so that patients are spending less time traveling to care, The average round trip to the oncology day ward in where I work is 120 kilometres in single car journeys in Cork. And also less climate toxicity, as healthcare is responsible for 5% of global healthcare emissions. But there's the generic slide for a meeting, but the patient reality for the meeting is very different. So you can see the top of the iceberg is what we think of for the patient, but there are the cost of toxicity for the patient is also more dramatic in terms of their home, in terms of potentially relationships from side effects of prolonged toxicity, in terms of financial burden of not being able to return to work, for instance, because they're getting a treatment performed. One of the barriers that we have identified in terms of de-escalation is the mindset of the doctor versus the mindset of the patient. So the mindset of, as this study from a French group that we're working on in the Optimisation trial, looking at the barriers to de-escalation trials and the facilitators in patients, you can see here that the physician mindset is very different to the patient mindset. And an added aspect of this is the stress of a cancer diagnosis, and I know that many of the patient advocates in the room will be able to relate to this, where you see the shock of the diagnosis followed by recovery, and this period is often the time that you're discussing with the patient about the role of a trial, potentially with de-escalation. They're terrified that the cancer is going to come back. They're concerned that you have a standard dosage approved by a regulatory body, and now you're looking at changing that and what the implications of that for them. The iceberg also applies in our system. So the iceberg in terms of financial toxicity relates to availability of new treatments. If you go back to the analogy of the antibodies getting together and having progeny, if we're spending all the money on the parents, we won't have money for the children. So we won't have money to allow for the try and buy and tetraspecific antibodies that probably will be as transformative in our environment as pembrolizumab and Herceptin in the past. So this is the data for pembrolizumab per event prevented. So this is per relapse prevented, which is almost $3 million per event averted. So if we can reduce that to half that, there are significant cost savings for society and for patients ultimately, because we will have more treatment options available for them. In reality, we've been doing this for a long time. So the first adjuvant trial for adjuvant chemotherapy for breast cancer was from Milan, and it was the CMF regimen, and we used to give 12 months of it, and then there was a de-escalation trial where we found 6 months was similar to 12 months. In Cancer Trials Ireland, we've also been involved in optimisation studies where new information with genomic platforms has shown that by using these platforms, we can predict patients who won't need chemotherapy. So work by Janice Walsh, led by Janice Walsh nationally, has shown that implementing the TAILOR-X trial results, which showed that you could safely omit chemotherapy for patients with early stage breast cancer, reduced chemotherapy prescriptions in this group by 53%. I think it's important to remember with the TAILOR-X and the RxPonder trial that in order to go on the trial, the doctor had to be convinced that the patient needed chemotherapy. In other words, that they could be randomized to chemotherapy. So these were practice-changing studies shown by the evidence guidelines on the right-hand side. At present time, we're also looking at other optimization trials, and this is the Optimal trial led by Marlene Koch globally and led by Roisin Connolly in Cork, where you're looking at tumor-infiltrating lymphocytes, which is a standard H&E technique on the cells, no genomic platforms involved, to see if some patients with triple-negative breast cancer don't need adjuvant chemotherapy. This would be a game changer for those patients. The radiation doctors have been at this for a long time, so this is one of the largest practice-changing studies called the Fast Forward trial, where we give 1 week versus 3 weeks of treatment. And the results of this became available around the time of the onset of the COVID pandemic in 2020, and practice changed overnight in many cancer centres as a result. The impetus for today came from this meeting in the Netherlands, led by the held at the Netherlands Cancer Institute, who've been involved in significant de-escalation trials, such as the SONIA trial shown on the right here. Studies were presented in a variety of tumour types, not just breast cancer, which is in the cartoon, and there was NCCP attendance, and we had met with the NCPE beforehand. and there was recognition for EU-wide collaboration to facilitate study conduct. One of the things about these de-escalation trials is you need to accrue quickly, and so the larger the network, the quicker the accrual, so that you get the opportunity to change practice before practice becomes embedded. Jarushka has mentioned the investigator-initiated workshop that was held, and this Sarah McLaughlin's work was featured by her, but I think it's important to emphasize that Sarah McLaughlin is productive and had 2 trials, not just one, at the meeting in patients with melanoma. Very pragmatic, straightforward trials, but with the potential to transform practice. These studies are not limited to hematologic malignancies, so this is a great trial called the Viper trial, and it comes from the Netherlands. And in this trial, what they're using pharmacokinetic adjusted dosing. So you can see that the drugs, venetoclax and ibrutinib, are given together. And by giving rabinavir in association with that, you optimize the drug, so you need less of the drugs. Hopefully they won't find out to make the 50 milligram and the 25 milligram tablet doses the same cost as a parent drug. But if in this case you can see dramatic drug cost savings in this cartoon. And our plan is with the HOVON group to introduce this in our lymphoma, in our hematologic DSSG. This is a trial that we want to look at, which is relevant to the patient that I discussed earlier. It's called the PLANET trial. And in the PLANET trial, patients who have surgery performed for triple-negative breast cancer after receiving the Keynote regimen are randomized to either chemotherapy alone or chemotherapy and pembrolizumab. And so the hope is for 1,000 patients, and we're looking at seeing ways in which we can get this study activated in Ireland. We've discussed it at our breast DSSG, and there's 100% buy-in by the clinicians involved. The cost of the trial is about 3,000 to 4,000 euros per patient for data management. The drug cost savings per patient randomized to the control arm is 70,000 euros. So it would be 35,000 euros for every pair of patients enrolled. This is a platform trial that we're developing in Cancer Trials Ireland in association with HACAM and the Breast International Group, looking at genomic platform integration. It's been looked at in low-risk node-negative disease, node-positive disease, and this is a more extensive node-positive disease subtype. It will be a global phase 3 study that we're trying to seek funding for at the moment. As Benjamin and the previous speakers have all outlined, funding is a significant sticking point in this. So one of the problems in many of our approvals is that we're looking at approve, escalate, approve, and then stop. So these de-escalation trials are like swimming against the tide. So how do we swim against the tide? So there are challenges for patients in terms of psychological and emotional burdens, equipoise and trust. Medical trust is hard-won and easily lost. Randomization anxiety, and then challenges for researchers in terms of equipoise maintenance, funding, and regulatory and payer inertia, as Benjamin has outlined. So what are the solutions to this? We're here to talk about solutions and not challenges. So you can see the patient-related ones are here in terms of patient integration in trials, and then there are research-related ones which are multifaceted, not just in terms of developing the study, but in terms of funder reform, in terms of smarter design, and in terms of regulation, and in terms of introducing new biomarkers, as was discussed earlier in the long proposal by David and Jarushka in Beaumont in KRAS patients. So how do I square the circle on all of this? So this is the square the circle is this. There are 4 key facilitators to this. There's public funding. There's early engagement of PPI, and that's why PPI is so important as part of this process. And I want to highlight that our Irish Cancer Society, Cancer Trials Ireland, Breast International Group, patient PPI project is being featured at ASCO, presented by Emer Mulvaney, and we're very pleased that there are so many patient advocates and patient partners on this presentation. A strong cooperative group infrastructure, not just in terms of Ireland, because we don't have the patient population to do the large practice-changing de-escalation trials that are needed here, but a global network, as Donald Brennan has shown in what they have done with the Precision Oncology Ireland initiatives, and in what we've been working on achieving in Cancer Trials Ireland in terms of our global network. And finally, a clinical champion, as Benjamin has been, to lead this. So thank you very much for your attention today.